In this video, I'm going to do just a brief kind of overview of all the different pathways that we went through, that we talked through, how they fit in with one another, and just kind of smoothing over all of the edges of everything that we've talked about so far. Now, this version of the pathways that I'm going to be looking over is the first one that I ever made, so it's a little bit neater than the one that I did in the videos, but it's the exact same. So we started with glycolysis, and I'm zooming in here. Glucose to pyruvate. This is everything in black. We go all the way down. Remember, there are the energy investment phase where we invested 2 ATP, and that goes all the way down to step 4 and 5. And then from here, remember that glucose splits from a 6-carbon molecule into 2 3-carbon molecules. We continue the pathway all the way down, and we end up producing 4 more ATP, so a net total of 2 ATP per glucose. And we end up here in the center with pyruvate, and this is going to be kind of our central hub because we can do a lot with pyruvate. Let's zoom back out. Everything in black, glucose down to pyruvate, glycolysis. Now, to get back up is gluconeogenesis. So that is everything in these dark blue. We started from pyruvate. Remember, some of them needed extra enzymes, but some of them, like enolase and all of these, they can go back and forth, and we don't need a separate enzyme because it's not a control point. And by control point, I just mean anything that is allosterically activated or inhibited. So for gluconeogenesis, remember we had those 3 different steps with the different enzymes. We have step 1, step 3, and step 10 are steps that need an extra enzyme for gluconeogenesis. And remember, our step 10 has this weird kind of intermediate in going from pyruvate back up to PEP. But as we learned, oxaloacetate is very, very important. Zooming back out, that was glycolysis and gluconeogenesis going up and down, glucose, pyruvate, pyruvate, glucose. Now let's focus on pyruvate. In the light blue, we can see anaerobic fermentation, and this is what happens when there's not enough oxygen in the body, but the body still needs to produce ATP. So the purpose of this fermentation is to regenerate NAD+. And if we remember, NAD+ is very important for glycolysis. We need NAD+ to be reduced to NADH for glycolysis to run and to produce more ATP. So the purpose of anaerobic metabolism is just to replenish the source of NAD+. So this first one going all the way to ethanol, this is what happens in yeast, not in humans. But the one down here that goes from pyruvate to lactate, called the Krebs Cycle, which is lactate back up to pyruvate, is what happens in humans. And the main purpose of the Krebs Cycle is to stop lactate buildup. Remember, if we have a lot of lactate, that can lead to acidosis, pH levels drop, not good. That happens when we exercise a lot. We're not getting a lot of oxygen to our muscles, but we still need to make ATP, so we're going to regenerate this NAD+ so that we can keep glycolysis going. And then we're going to turn lactate back into pyruvate to avoid that pH drop. Now, lactate dehydrogenase, remember there are 2 different kinds, one in the heart, one in the skeletal system, different activators, inhibitors for each. Zooming back out. We covered anaerobic fermentation over on the right, and now we're going to go down to the transition step, which is this in green. The transition step is what gets us from pyruvate to acetyl-CoA. And remember, there's something called the pyruvate dehydrogenase complex, and those are these 3 enzymes that make this happen. We're also losing a carbon dioxide here, but it gets us to acetyl-CoA. Now, we need this in combination with oxaloacetate from gluconeogenesis, or as we'll learn later, it comes into a lot of places in order to kick off the citric acid cycle. So the citric acid cycle is everything in this dark green color. We're going to follow it down, and the main purpose of the citric acid cycle is to produce these highly reduced molecules like NADH, FADH, to be sent to the electron transport chain to make more energy, make more ATP. It also produces GTP, which can also be turned into ATP, more energy. So the main purpose of this is just energy production. So we go all the way down, we follow the green all the way down, and we end again at oxaloacetate, coming into play again. Oxidative phosphorylation is the process in which we form ATP, electron transport chain, all of that goodness. Zooming back out, we can see all of the allosteric effectors. This is everything in orange. So everything in orange is going to be what affects these enzymes. So most of the time, if there is a high level of a product of an enzyme, that's going to inhibit it. Because if there's a lot of product buildup, then why would we want to activate the enzyme to keep making more? So it's mostly going to be a negative feedback mechanism there. Let's look at the fatty acid synthesis. It's going to take the citrate from the mitochondrial matrix. If you remember, the citric acid cycle is in the mitochondrial matrix, and it's going to transport it to the cytosol. Turn it into oxaloacetate, acetyl-CoA. We'll learn a little bit more about this later, but that is how fatty acid synthesis kind of fits into all of this. Oxaloacetate, if you remember, can be turned back and forth into malate here and here. And from here, it can be turned back into pyruvate. That's how fatty acid synthesis kind of fits in. We can scroll down to the blue, which is the glyoxylate cycle. This kind of has to do with carbohydrate synthesis. It's kind of a side step of the citric acid cycle that bacteria and plants do. And the main goal is just to kind of produce some of the intermediates of gluconeogenesis. So we move isocitrate over, we grab succinate, and we are forming malate, oxaloacetate, and eventually that's going to go to carbohydrate synthesis, and that is a pathway for another day. Zooming back out, we finished everything down here at the bottom. We're going to scroll back up. We're passing through our transition step, passing through our anaerobic fermentation, pyruvate hub down here, and we're climbing all the way back up to the top of glycolysis. Now, we can focus on glycogen and how that plays into everything. Remember, glycogen is the storage of glucose. So glucose gets stored as glycogen when we're not using it. So glycogen breakdown is going to happen when we don't have enough glucose. Glycogen breakdown is this dark purple, and if you remember, it turns into glucose 6-phosphate, and in the muscle, it can go directly into glycolysis from glucose 6-phosphate, completely skip that first step. But in the liver, glucose 6-phosphate has to be turned back into glucose before it can enter glycolysis. Glycogen synthesis is the opposite. We're going to turn glucose into glycogen, and this is going to happen when we have too much free glucose and we need to store it. So it's going to have the opposite effectors as glycogen breakdown does, but different enzymes. We can scroll over here. The orange is the different kinds of forms of these enzymes, and then the blue-green color is going to be the hormonal control. So remember, glycogen phosphorylase has an active form and an inactive form. Phosphorylate between the two to get to one to the other. Same with glycogen synthase. There's an active form and then an inactive form. Phosphorylate to get in between the two. And then the main hormones that affect glycogen breakdown and synthesis is glucagon and insulin. Glucagon, which I remember as glucose gone, that's going to promote glycogen breakdown. Don't have enough glucose, we're going to activate those enzymes that break down glycogen to give us more glucose. Insulin, on the other hand, insulin is secreted when we have a lot of glucose in our blood. We just ate a big meal. So insulin is instead going to activate the enzymes that promote glycogen synthesis. So we have a lot of glucose, we want to store it. So that's the hormone response. Very lastly, we very briefly touched on the pentose phosphate pathway. This is just a way to produce NADH, another highly reduced molecule, to eventually produce more ATP, and also to produce these ribose sugar bases that can be used for nucleotide synthesis. And also over here in this blue all by itself over here is the fructose 2,6-bisphosphate and how that can regulate step 3 of glycolysis. So that is our entire flow map. It seems like a lot. Go through the videos, try to get a grip of it, but I promise writing it out itself makes it stick in your head a lot better. I will post this whole thing on the link tree. It will be under biochemistry notes, and then all biochemical pathways is what it'll be called. But yeah, it's easier to visualize now how everything fits together, the different control mechanisms. So that is all I have for you today, and that kind of wraps up our biochemical metabolism. Yeah.